Hello everyone, today we will be covering plant metabolism in this lecture. This involves photosynthesis, including light and dark reactions, and C3, C4, and CAM plants and their pathways. And we'll also go over some interesting primary and secondary plant metabolites, as well as a bit at the end about isotope applications that are not isotope tracing. So let's get started. So first, I'm going to go over some basic plant structures that you have probably learned in your earlier classes. So one of the major structures involved in a lot of plant metabolism is the chloroplast. So the chloroplast interior is called the stroma, and there is also the thylakoid, which are membrane brown discs inside the stroma. And these discs are made out of thylakoid membrane. And there is also the structure called stromata, not to be confused by the stroma, which are two completely different things. So the stromata are these little air holes on the surfaces of plants that is used for gas exchange. So this is what they look like when they're closed, and when they're open, they look like this, and gas can enter and is released out of the plant. So today we'll be going over photosynthesis. So photosynthesis is basically the opposite reaction of cellular respiration, which we covered extensively in this course. So photosynthesis takes place in the chloroplast, more specifically in the stroma and also the thylakoid lumen. So photosynthesis involves two parts. First part is the light reactions, which transforms light energy into reducing power, aka NADPH, and ATP. So this light reaction occurs in the thylakoid membrane, while the dark reactions, also called the Calvin cycle, uses the light reaction products in order to drive the reduction of inorganic carbon or carbon dioxide into sugars. So, and this occurs in the stroma. So as I said previously, light reactions occur on the thylakoid membrane. So on these membranes are photosystems 1 and 2, which absorbs light and converts that into chemical energy, such as ATP and NADPH. So more specifically, photosystem 1 and 2 would. So more specifically, photosystem 2 first absorbs photons and converts water into O2 and protons and electrons. So these electrons then travel to the cytochrome 6bF, which pumps protons from outside into the thylakoid lumen. And then finally, at PS1, the light is able to convert the NADP plus into NADPH using a reductase. Now, because the proton gradient was formed using the cytochrome B6F pump, there's a higher concentration of protons inside the thylakoid lumen compared to the stroma, which fuels the synthesis of ATP using ATP synthase. So this process is almost the exact same as the ATP synthase reaction in the electron transport chain in the mitochondria. Essentially, the concentration gradient of protons generated would generate proton motive force, which would allow ATP synthase to synthesize ATP from ADP. Now, chlorophyll is also an important component to the light reaction. So chlorophyll are in the photosystems, and they are the ones that are absorbing the light in the photosystem. So they do this because they have this extensive resonance structure, which allows them to absorb light, and different chlorophyll, for example, chlorophyll A and chlorophyll B, absorb different wavelengths depending on their chemical properties. And this facilitates the conversion of that light energy into chemical energy. Now, interestingly, chlorophyll is a sign that potatoes are poisonous. So whenever you see a green potato, please do not eat it, because that means that the potato at its current state is poisonous. So the reason behind that is that chlorophyll production in potatoes indicates that light reactions are occurring. And light reactions activate toxin synthesis in potatoes, generating these toxins that inhibits acetylcholinesterase, which is an important enzyme in our bodies that regulate nerve impulses. So another interesting thing about chlorophyll is that during autumn, chlorophyll molecules start to degrade in plants. And this leaves behind these carotenoids molecules, which are brown, which causes the change in colors from green to brown/orange/red. And carotenoids are brown simply because they absorb a different wavelength than chlorophyll molecules. Now we're going to go over the dark reactions, or the Calvin cycle. There are 3 stages to the Calvin cycle: the carbon fixation stage, the reduction stage, and the regeneration stage. And this all occurs in the stroma. So in stage 1, carbon is fixed into an organic form. So this whole step is catalyzed by an enzyme commonly called Rubisco. So Rubisco is a very slow enzyme, which is why this step is the rate-limiting step of the Calvin cycle. So in this step, inorganic carbon, as the form of carbon dioxide, is fixed into an organic form and eventually is converted into two free phosphoglycerates. Now, Rubisco is actually the most abundant enzyme in the world, which is interesting because it is so inefficient. But actually, this is why it's the most abundant enzyme in the world, because Rubisco is very inefficient, fixing only 3 carbon dioxides per second, which is extremely slow for an enzyme. And to compensate that, plants would have to make a lot of Rubisco, which is why a huge percentage of plants is actually just made of Rubisco, which is super cool. In fact, around 2.5% of the global mass of leaves are due to Rubisco. And Rubisco actually has this wasteful side reaction that makes it even more inefficient. So Rubisco can use oxygen instead of carbon dioxide in its reaction, forming a free phosphoglycerate, but also a phosphoglycerolate. So phosphoglycerolate can be salvaged, the carbons can be salvaged from it, but ultimately for this reaction, you are losing a net carbon as a CO2 because CO2 doesn't enter this carbon fixation step. So this oxygenase reaction is also called photorespiration. And photorespiration is a huge problem when the temperature is hot, because during hot temperatures, Rubisco has a decreased specificity for CO2, and also CO2 solubility into the leaves is lowered. So during high temperatures, Rubisco is less sensitive for CO2, so it's more likely to go through this photorespiration wasteful side reaction rather than the useful carboxylase reaction that we saw previously. Now, step 2 is the reduction step. So in the reduction step, the two free phosphoglycerate that were formed previously is ultimately converted to these hexose phosphate sugars. And it does this by utilizing ATP and NADPH that were originally made from the light reactions. So these monosaccharides can be used to form more complex sugars, such as sucrose, starch, and cellulose. So starch is a major sugar in plants. It is stored in the chloroplast and comes in two different forms, the amylose and the amylose pectin, depending on what type of linkages are formed between the monosaccharides. And then finally, the third step of the Calvin cycle is the regeneration step. So after all of that, after generating the sugars, you need to regenerate ribulose 1,5-bisphosphate. In order to regenerate this compound, you need these PPP metabolites, ribose 5-phosphate and cellulose 5-phosphate, in order for more carbon fixation to occur in the cycle. So this is just an overview of the Calvin cycle. Ultimately, what you should know that the first step is the step here where inorganic carbon, or CO2, enters the Calvin cycle and produces free phosphoglycerate. Free phosphoglycerate is ultimately formed into hexose sugars via the consumption of ATP and NADPH. The PPP metabolites are used to regenerate ribulose 1,5-bisphosphate so that the cycle may continue. All right, so the Calvin cycle is intimately linked to the light reactions simply because it depends on the light reactions products in order to occur. So the light reactions leads to favorable conditions for the Calvin cycle to occur in the stroma. So how it does this is during the light reactions, cytochrome B6F pumps protons out of the stroma into the lumen, leaving the stroma at a lower concentration of protons or in an alkaline state. And because of this, magnesium ions leak out of the lumen into the stroma to compensate for the deficiency in positive charge caused by this cytochrome pump. And because of these two conditions happening, Rubisco is able to be activated. So the activation of Rubisco requires two steps. The first step is where it produces this proton here. So this step is more likely to occur in alkaline conditions. And then in the second step, it requires a magnesium ion in order to be fully activated in this form, which is why when the stroma has low protons and high concentrations of magnesium ion, it is favorable for the Calvin cycle to occur. So another thing that we briefly touched on is that the light reactions produce an NADPH in the stroma in order to fuel the Calvin cycle, specifically that reduction step in the Calvin cycle. Now we're going to get into some different types of plants and the different ways that they fix carbon. So C-free plants are pretty generalized. They use the Calvin cycle as we saw it, previously. So there's no deviations from this Calvin cycle. Meanwhile, C4 plants have a slightly different way of fixing their carbon. So the carbon fixation process is a bit longer and requires both mesophyll cells and bundle sheath cells. So what happens is that the carbon enters the mesophyll cells and is used to carboxylate PEP into oxaloacetate, and then oxaloacetate gets converted to malate. Malate becomes a carrier for carbon, and so malate transports this carbon into the bundle sheath and then dumps the CO2 for the Calvin cycle to occur. Now, this pathway is called the Hatch-Slack pathway. And the purpose of this pathway is to transport CO2 into the bundle sheath cells, so more interior located cells, in order for CO2 to be concentrated in these cells and allow for the Calvin cycle to occur more efficiently. So the Calvin cycle occurs more efficiently when CO2 is concentrated there because it accelerates the carboxylase reaction over the photorespiration reaction in the carbon fixation step of the Calvin cycle. This means that C4 plants are extremely well adapted to hot climates because it is able to overcome the increased photorespiration that occurs naturally in hot climates. Now, CAM plants are a special type of C4 plant that separates carbon dioxide accumulation and utilization temporally rather than spatially like other C4 plants. So instead of having the accumulation in the mesophyll and then the utilization of the CO2 in the bundle sheath cells, CAM plants first consume CO2 and carboxylases it into malate during the evening, and then utilizes the CO2 from malate in the Calvin cycle during the day. And this all happens in the mesophyll of the CAM plants. So the reason why they have adapted to do this pathway is because CAM plants are adapted to live in hot and dry environments, which means that they want to conserve water. So to conserve water, they only open their stomatas during the evening when it's cooler. So gas exchange only happens in the evening, therefore they only get CO2 entering the mesophyll at night, and CO2 accumulation can only happen at night, and then during the day, the CO2 is used in the Calvin cycle to generate sugars. So this is why CAM plants are slow growers. It's because their malate storage is limited, so they can only produce a certain amount of sugars during the day. Now we're going to go over some plant primary and secondary metabolites. So primary metabolites are mostly the ones we've learned in this course, those that are involved in basic life functions such as growth, development, and reproduction. Now, and secondary metabolites are metabolites that assist plants in surviving in their environment, whether it's defending against predators or signaling in symbiotic relationships. Now, this first plant primary metabolite we're going to go over is MSG. MSG, you've probably heard of it. It is the flavor enhancer that gives an umami taste. It was originally isolated from seaweed, and interestingly, it is chemically indistinguishable from glutamate. So MSG is just made out of a glutamate and a Na plus ion. Unexpectedly, it's commonly believed that MSG is harmful, which is odd because MSG is made out of mostly glutamate, and glutamate is one of the most abundant amino acids in our bodies. So there is actually no chemical reason why MSG would be harmful to our bodies, and there's also no compelling evidence that MSG has any adverse effects after consumption. So the next secondary metabolite is flavonoids and isoflavonoids. These molecules are involved in ligand rhizobium interactions. So as if you recall in our nitrogen fixation lecture, we learned about the symbiotic relationship between ligand and rhizobium bacteria. So what the signaling factors do is they're produced by the ligand plants through the roots. And then these flavonoids and isoflavonoids signal the bacteria to produce node factors. These node factors are then used to signal the plant roots to undergo the formation of nodules. These nodules are the site of infection for the bacteria. So essentially where the bacteria would reside in the roots in order to do its nitrogen fixation and help the plant grow. Resveratrol is the next secondary plant metabolite we're going to go through. So it is produced by plants such as grapes and cranberries and peanuts in response to injury or infection. So the proposed cardiovascular health benefits of wine is actually attributed to the presence of resveratrol in wine. So resveratrol is thought to prevent damage to blood vessels, reduce bad cholesterol or LDL cholesterol. However, there is very little evidence to support these claims, much like other dietary supplements that we have learned in this course. So resveratrol is biosynthesized in base of phenylalanine and tyrosine. So these two metabolites are made from aromatic amino acid biosynthesis in plants or grapes, and then they can branch off and make resveratrol. So capsaicin is a plant secondary metabolite produced by a lot of fruits and vegetables and is the ultimate cause of spiciness. It is produced as a defense mechanism against mammals and fungi so that they do not consume the plant. And it is produced from phenylalanine and valine. And this is what it looks like. So just as for your interest, the Scoville heat units is a measure of spiciness, and the concentration of capsaicin in the plant is what determines its SHU value. So here you can see bell pepper has a SHU of 0, and all the way up here, the Carolina Reaper has a SHU of 2 million, which makes it probably one of the most spiciest vegetable there is. So now we're going to switch topic and talk about some applications involving isotopes. So first, we're going to talk about the kinetic isotope effect, which is when a heavier isotope causes the slowness of a reaction, slows down a reaction. So generally, the reason behind this is because heavier isotopes have a higher activation energy in most reactions compared to lighter isotopes. So the photosynthetic reactions are an example of this. So both Rubisco and phosphoenolpyruvate carboxylase are slowed down by 13C isotopes as their substrates. So, which means that Rubisco and PP carboxylase both prefer the lighter isotopes, so 12C isotopes, rather over the 13C isotopes. However, different enzymes have different levels of preferences for 12C. For instance, here PP carboxylase prefers PEP carboxylase is less selective for 12C than Rubisco. And because of this, the 13C signature in C3 and C4 plants are significantly different. So the measure of 13C signature usually is written as delta 13C and the units are per mil. So this is odd, but the higher the negative per mil value, the less 13C isotope signature is in the sample. So in this case, in this case, C3 plants have a lighter 13C signature than C4 plants. So the reason behind this is, as we said before, PEP carboxylase is less selective for 12C. Hence, since PEP carboxylase is the first enzyme that uptakes carbon dioxide from the air, C4 plants have a higher concentration of 13C, takes up a higher concentration of 13C carbon dioxide over C3 plants that uses Rubisco, a more selective enzyme. And now this kinetic isotope effect can be used in many ways. One of the ways it can be used is to determine the plant-based diet of an animal based on if they ate, based on their 13C signatures. So if the 13C signature is higher and between -16 to -12, then it's more likely that the plants, the animal has eaten C4 plants rather than C3 plants. Now, the other use for isotopes is carbon dating. So carbon dating uses 14C isotopes rather than 13C or 12C. So here, radioactive 14C CO2 is constantly being made in the atmosphere and then is absorbed into living organisms. And then based on the 14C signature, we can determine how old an organism is because 14C decays slowly over time. So the less 14C there is in a sample, the older it would be. In fact, 14C is especially useful for this because it decays extremely slowly with a half-life of around 6,000 years, which means that using this method, we can date organic samples that are very, very far into the past. So an example of carbon dating is when scientists dated the CO2, the 14C CO2 that was trapped in the polar ice sheets. So using the 14C signature, they were able to determine the date at which the ice age ended. So the ice age was dated to be ended around 12,000 years ago, based off of carbon dating. So another application of carbon dating is we actually used this method in order to date the age of Jesus's shroud. So unexpectedly, we determined the age of the shroud to be around 600 to 700 years ago, which doesn't make much sense considering that the shroud should have been around 2,000 years ago. So here are the learning objectives for this lecture. You should know the basic plant structures that are associated with metabolic pathways, the light reactions and the dark reactions, and the C3, C4, and CAM plants and the ways that they fix carbon, and also some of the plant primary and secondary metabolites, as well as the applications of isotopes.